In this video, I will focus on the molecule ATP, adenosine triphosphate, and how the hydrolysis of its terminal phosphate results in the release of energy that can then be used to fuel other biochemical processes. I will also cover some key AP Bio exam questions that center around this biochemical energy currency. But let's get started with ATP. Why does ATP hydrolysis result in the release of energy? Let's take a look at the composition of the ATP molecule. It is composed of an adenine group, which is basically a nitrogenous base. It also has a 5-carbon ribose sugar and 3 phosphate groups, all attached in one molecule. There are 2 key bonds, one inside adenosine triphosphate and another bond inside water, that with the appropriate amount of kinetic energy can be broken. And when new bonds are formed between hydrogen and phosphate and the releasing hydrogen ion, stronger bonds are made and therefore a lot of energy is released. It is actually because of this, the breaking of weak bonds and the forming of stronger bonds, that ATP is considered to be such an amazing energy currency. Check out my video in which I model with my students the breaking and making of bonds. How does creatine phosphate provide energy to the muscle? So there's another system that involves a phosphate group. In this case, the phosphagen system, which centers around the enzyme creatine kinase. Here's how it works. Phosphocreatine contains a phosphate group, as the name phospho implies, and creatine kinase is capable of removing that phosphate group and transferring that phosphate group to ADP, adenosine diphosphate, thereby creating ATP, adenosine triphosphate. And as a result, Popeye is now able to lift so much more mass because of all this power given to him. How does ATP hydrolysis compare to the phosphagen system? So let's take a look here. We've got 2 axes going on. On the x-axis, the reaction coordinate, and on the y-axis, Gibbs-free energy. Let's take a look first at the phosphagen hydrolysis that we just talked about. You see here an exothermic graph. Why exothermic? Because it shows you that the end of the graph is lower than the beginning of the graph, because more energy is released as a result of making stronger bonds than the energy that was required to break the original bonds. Here you see again the phosphocreatine molecule with the phosphate group that is removed, resulting in creatine by itself, and that releases 43.1 kilojoules per mole. How does that compare to ATP hydrolysis? Again, ATP hydrolysis, where we're starting with adenosine triphosphate and resulting in adenosine diphosphate with a concomitant energy release of only 30.5 kilojoules per mole. So comparing the two, the phosphagen system does release more energy when comparing it to ATP hydrolysis. What is the output difference between the phosphagen system, the glycogen-lactic acid system, which I'll cover in a sec, and aerobic respiration? So if we first look at the phosphagen system, we see that it only lasts about 8 to 10 seconds, gives a ton of power, but it's completely dependent on the presence of phosphocreatine in the muscle. And there's not that much there. The glycogen-lactic acid system is also dependent on energy storage in the muscle and in the liver to a certain extent. And that is namely glycogen, which is, as you may remember, a chain of glucose linked together. When that glycogen is hydrolyzed, you basically have glucose monomers that can then be put through glycolysis, and in the absence of oxygen can give rise to lactic acid and a little bit of ATP. That storage gives rise to energy for about 1.3 to 1.6 minutes. So how does this then compare to aerobic respiration? Well, aerobic respiration, as the name implies, requires the presence of oxygen, because oxygen is the final electron acceptor in the mitochondria, at the end of the electron transport chain. But this is a slower process, the advantage of it being that it lasts much longer than either the phosphagen or the glycogen-lactic acid systems. We are now ready to start with our AP biology questions. Let's start with this first one. Muscle contraction depends on ATP hydrolysis. During periods of intense exercise, muscle cells rely on the ATP supplied by 3 metabolic pathways: glycolysis, mitochondrial respiration, referred to earlier as aerobic respiration, and the phosphagen system. Figure 1 shows the rates at which the 3 metabolic pathways produce ATP following the start of an intense period of exercise. The question is now as follows. Based on the provided data, justify the claim that the phosphagen system is an immediate, short-term source of ATP for muscle cells. If you look at the graph highlighted in the diagram, ATP production from the phosphagen system is maxed out in the first 10 seconds of an intense period of exercise, very similar to like a HIIT workout, high-intensity interval training, with ATP production topping at 43 kilocalories per kilogram per minute. Glycolysis, on the other hand, maxes out at about 20 seconds. Finally, mitochondrial respiration keeps going strong, even after 100 seconds of intense exercise. So the curve doesn't really drop off. It keeps rising slowly. Obviously, it never peaks like the other two systems. So we just went over the graph that you're seeing here on this slide, but this question really is a multiple choice question. And we're answering the same question. Based on the provided data, justify the claim that the phosphagen system is an immediate short-term source of ATP for muscle cells. Let's check out option A. ATP production by the phosphagen system increases and decreases rapidly following the start of the exercise period. Pretty much exactly what this graph shows. Sounds like a pretty good choice to me. Option B. ATP production by the phosphagen system increases gradually. I'm going to pause right there because clearly this quick peak does not match gradually. So not a good choice. C. The ATP produced by the phosphagen system contains more energy per molecule than does the ATP produced by the other pathways. I'm not sure if this is correct or incorrect, this statement, but does it answer the question? I don't think so. D. ATP hydrolysis in muscle cells occurs immediately after the start of the exercise period, but stops before the end of the exercise period. Again, does this choice actually answer the question? So let's take a look at the key. Option A is our best option. Next question. The following question shows you a graph with 4 curves labeled number 1, 2, 3, and 4. And the following question. Which of these 4 reactions, as represented by the 4 curves on the right, can be coupled to make the desired products? So that question in and of itself might be a little bit confusing, but it basically underscores exactly what is happening during ATP hydrolysis. Energy is released, and that amount of energy can then be used, can then be coupled to a different reaction that requires energy. So let's see how we can apply that concept to this question. So the first graph, number 1, the reactants have a relatively high amount of chemical potential energy. But once this reaction has been completed and the products have been formed, the products have a much lower amount of chemical potential energy, showing you that this reaction is exothermic, as represented by this double arrow and the products being lower than the original reactants. Similar for the second curve. Again, the energy of the reactants are much higher than the energy of the final products. Again, this is an exothermic reaction resulting in an energy release. The amount of energy released by the second reaction is slightly less than the first reaction. But now we should be able to answer the question by looking at curves 3 and 4. If we look at graphs 3 and 4, we see a different pattern. 3 and 4, the products have more chemical potential energy than the original reactants. So graphs 3 and 4 illustrate reactions that are actually endothermic as opposed to exothermic. Now, if we look at the two double-headed arrows, the one for reaction 3 is much larger than the one for reaction 4. And now we can actually read this text here. Reactions 1 or 2 can be coupled to reaction 4, but not to reaction 3 for exactly this reason. The energy required to make reaction 4 happen, as indicated by this smaller double-headed arrow, is less than the energy released by either reaction 1 or 2. The energy required to make reaction 3 happen is much larger than can be provided from the energy released by reactions 1 or 2. This question revolves around the enzyme hexokinase. Let's first read it. The enzyme hexokinase catalyzes the conversion of glucose to glucose 6-phosphate, which is an important step in glycolysis. The reaction involves the transfer of a phosphate group from ATP to glucose. I'm going to demonstrate that in a sec using some models. Either a glucose molecule or a water molecule can fit in the active site of hexokinase. The presence of a water molecule in hexokinase's active site would result in the hydrolysis of ATP to ADP instead of the conversion of glucose to glucose 6-phosphate. Which of the following statements best helps explain the reaction specificity of hexokinase? So let's first take a closer look at the actual reaction. You're seeing here a model of the hexokinase enzyme and the glucose molecule. And then you see the molecule ATP, again, adenosine triphosphate. Hexokinase facilitates the transfer of the phosphate group from ATP to glucose, thereby turning glucose into glucose 6-phosphate. And that conversion can happen because of an induced fit. When glucose sits in the reaction site of the enzyme somewhat changes its conformation to better fit that interaction, thereby promoting the production of glucose 6-phosphate. Let's go back to the question now. Okay? Which of the following statements best helps explain the reaction specificity of hexokinase? And I'm taking you straight to the correct answer. D, glucose has the right shape and charge to cause hexokinase to undergo a structural change. I referred to that in the previous slide as an induced fit. So this is better and has been tested by experimentation as the lock-key model of enzyme substrate. So this induced fit, this structural change, is needed for catalysis, whereas water does not. I hope that answers this question without going through the other 3 choices. Next question involves ATP again, but now we're in chloroplasts. ATP is synthesized from ADP plus inorganic phosphate in a reaction catalyzed by ATP synthase molecules that are embedded in the thylakoid membrane, again inside the chloroplasts. Which of the following statements provides evidence to support the claim that no ATP will be synthesized in the absence of a proton gradient across the thylakoid membrane? Let's check out that system again. Okay, so what we're looking at here is the thylakoid membrane. So north of that membrane in this diagram is the thylakoid space. South of this membrane is the stroma, which is inside the chloroplasts. Embedded in the membrane is the ATP synthase molecule. And the ATP synthase molecule is capable of funneling these protons through the ATP synthase itself. And because that is a favorable movement due to the high concentration of protons inside the thylakoid space, that basically results in energy being released that can then be used to turn ADP and inorganic phosphate to ATP. Let's now go back to the question. So, which of the following statements provides evidence to support the claim that no ATP will be synthesized in the absence of a proton gradient across the thylakoid membrane? Let's immediately go to the correct answer. No ATP is synthesized when channel proteins that allow the free passage of protons are inserted into the thylakoid membrane. So this would mean that instead of the hydrogen ions going through ATP synthase, channel proteins would allow these hydrogen ions to go through where they shouldn't go through, where it wouldn't result in the production of ATP. And that's why choice C is the correct answer. Let's take a look at choice A, which was another favorable choice, and why it's not correct. Blocking electron flow from one carrier to the next in the electron transport chains blocks formation of a proton gradient in the thylakoid. That's an interesting choice, but why is it not correct? Choice A does not link the proton gradient to ATP production and is therefore not the best answer. So I've now taken you through ATP hydrolysis and several AP biology exam questions. I hope you took away a lot of useful information for the AP Bio exam. Thank you for watching. Bedankt voor het kijken. En tot ziens. See you in the next video.